Brief introduction about myself. My name is Hu Bing Zhao, currently a lecturer at University College London. I'm the chair of this afternoon session. So in this afternoon, we have like 6 speakers in total, and for each presentation, it will be about 20 minutes. And so we usually the presentation will be around 16 to 80 minutes plus 3 to 5 minutes for question. Okay, so now let's welcome to our first speaker, Dr. Juan Liu, who is currently a postdoctoral researcher at Chinese Institute for Brain Science at Beijing, China, and talking about genetically encoded neurotransmitter sensor in brain research. Welcome.
It's my pleasure to introduce my part work here today to better fit our this seminar's topic of bioimaging. So I choose this topic is about genetically encoded neurotransmitter sensor in brain research. And so what is genetically encoded sensor? Genetically encoded sensor uses an interdisciplinary approach, and gene-coded chemical probes have been developed to visualize biological phenomena such as neuronal signaling in living cells. These optical tools can non-invasively label cells and record their activity with high spatial and temporal resolution. And so this is with the high sensitivity and specificity. It means that when it even have a lower neurotransmitter, and then it will also detect it could show the directly image by the microscope, and then with a high spatial temporal resolution, it also is the third with the encoded sensor, and of course it's too easy to use. And then I will introduce for you that what is the neurotransmitter. Neurotransmitter is a signal molecule secreted by neuron or glial cells. It affects another cell across the synapse. And like here, you can see this cell, this red body, that is the neurotransmitter. And it will be released, and it will be released with the presynaptic terminal, and if he gets the signal, and it will be contacted with the membrane, and it will be released with this gap junction. And then the post-synaptic neurons, it will be then gets the receptor will catch this neurotransmitter, and then the signal it will transfer to the other mechanism for the for the other activity. And then for today, I will introduce the different genetically encoded sensor with the neurotransmitter. And so the first, that is Dr. Jing. That's where I'm working. I'm doing the postdoctoral Dr. Jing's lab, and he explored this acetylcholine sensor. What is acetylcholine? Acetylcholine regulates a diverse array of physiological processes throughout the body. Despite its importance, chronological transmission in the majority of tissue and organs remains poorly understood, owing primarily to the limitation of available acetylcholine monitoring techniques. Acetylcholine broadly applicable tool for monitoring chronological transmission underlying diverse biological process. So they insert the CBGFP to this GPCR, and when it have the acetylcholine, it will have the signal with the it can detect this signal. And this sensor is with the delta F to the F0 is have the have nearly 9% compared to the first they explored the first version, and then they optimized the second version, and then they if it work, they don't know, so they applying them into the cell culture with the neuron, and it's also with the acute slice, acute brain slice, and also did the viral expression of the in vivo of the acetylcholine 2.0 for 6 hours, and after that they detected it under the 2-photon microscope with the give the mice with the visual stimulation, and found that all these they after that found that the acetylcholine 2.0 have a higher had a higher higher response for compared to the first version, and this work also published at the Nature Biotechnology in 2018. And after that, they did the optimization again. It's with they found that with this acetylcholine with the 3.0, they used the strategy with the linker optimization and used the CpGFP optimization. And found that, and then after this CpGFP application, when they have to captured with a neuron, they detect with the difference with the cell body and the axon and the dendrite, and with the even like this small spine, and found that this the third version is have the higher, is nearly 3 times compared to the before they explored the sensor. So work in the sometimes the sensor work good in the culture cell, but it not means it's very good can good in the can working good in the in vivo. So they did the accurate slice and found that is increased 0.7. So and after 70%, and then after they also injected with the HC, especially expressed this sensor as the acetylcholine sensor to the neuron, and then to did with the different behavior with the mice, and then recorded with the 2-photon microscope, and found that even the mice is running, and then the acetylcholine have a higher release when it's running and compared to when it's asleep, and there is low acetylcholine to show that. And so this data they published in last year with on Nature method, and so this is the improved sensor together other GPCR-based neurotransmitters sensor recently developed should help to address the fundamental biological questions on neuromodulation. I will introduce the second genetically encoded sensor, that's the dopamine. What is dopamine? Dopamine is a central monoamino neurotransmitter involved in many physiological and pathology processes. In the mental central nervous system, dopamine regulates a wide range of complex processes, including reward signaling and reinforcement learning, attention, and motor control. And we know that in the human brain, impaired dopamine transmission is associated with neurological disease, including ADHD, schizophrenia, and Parkinson. In addition, psychosis stimulants like cocaine act by acting the extracellular dopamine level in the brain to access. So Dr. Li's lab, they report the development of this GPCR, this dopamine sensor. They used the same strategy and found that this GPCR were engineered by coupling a conformational sensitive secure permeated EGFP and to select a human and dopamine receptor. So after that they also passed this application in a fly and zebrafish and the mice. And this is their data with the slice. And they found that with the dopamine, with the middle apparent affinity to the dopamine, and it have a lower response, and is with a higher another sensor is if with a higher apparent affinity, it will show the higher if in a different electrode stimulation and have a lot of dopamine release. And after that, they they did a because this is working. This sensor working with the with this green fluorescence. So, but to want to make it more, and so they explore this red dopamine again. So that is they have published a different in different paper with in 2018 and 2020. And so as an important then monoamine neurotransmitter, then have another one that is norepinephrine. Norepinephrine is very similar with the dopamine. The structure, you can find that the difference with the dopamine here is only have one hydroxyl group here. So they have the same similar structure. So they also have the same similar functions. So if you first when they do this sensors, they found that when it also can detect with the norepinephrine, and so they explored another sensor that is especially with norepinephrine. So they optimized with the different strategy and found that it have the much higher. It's nearly 1,000-fold higher affinity for this norepinephrine compared over the dopamine sensor when expressed in the neurons, which is an important tool for studying the norepinephrine-related neuron circuit. And after that, they also Dr. Li's lab also found that with another serotonin. Serotonin is also a very important transmitter in the brain research, and so serotonin signaling in brain plays a critical role in a wide range of including mood control, reward processing, and sleep-wake homeostatic regulation. Indeed, actually, as drugs targeting central serotonin activity have been used to treat virtual every psychology disorder, with the best example being the use of the selective serotonin. And so they explored this sensor when it meets with the serotonin, and the inactive state will change to the active state and optimize the insection. First, they optimize the optimization of the change the insection site, and then the second, the linker optimization, and then the third step, the CBGFP optimization, and then is over like nearly the variant is nearly over the 1,000. And when they showed the experiment, that is the serotonin when the basal is only can very lower influence with the captured neuron, and then when add the 10 micromolars serotonin, it will show that it's much higher signal. And if you use with the mutant tension, mutant sensor, then there is low signal. So that means so this is worked with the. So this signal is worked by this serotonin sensor. It's not the cell's spontaneous signal. After that, so this is they published last year also. And this year they found that this we know that they developed another sensor is very important, is endocannabinoid ECBS. We know that ECBS are retroactively neuromodulator with important function in a wide range of physiological process. The biological function of ECB is primarily mediated by the activation of the type 1 and the type 2 cannabinoid receptor. That is CB1R and CB1 receptor and the CB2 receptor. And mainly these two NIP and with this ECBS and is mainly two NIP, the metomibin is one is 2AG and another is AEA. And in the brain, and ECBs participated in short-term and long-term synaptic plastic of glutamatic and GABA energical synapses in a variety of regions. So including the so this is will express including the cerebral cortex and the hippocampus and the striatum and the ventral temporal area and amygdala and the cerebellum is different area in the brain, and so plays an important role in a wide range of physiological processes such as development, emotion state with pain, and the sleep-wake cycle, and the energy metabolism, reward, and learning and memory. So, and for this sensor, they have with the collaborator and also published this year. They found that in the Yuan Slaughter's laboratory uses this sensor progress to illustrate the molecular characters and the spatial dynamics of cannabinoid signaling in the hippocampus. In the hippocampus of living animals, they found that is the seizure, the epilepsy, elicits a great release of 2AG, and this which provides a substrate for long-term stroke-like symptoms compared with the normal physiological activity. So that means so the sensor is not just a sensor; it can work and can help the neurological scientists and so to do the research and can help to do a lot. And I will introduce this sensor because we used this sensor with the ATP sensor, and the adenosine and triphosphate is a universal. ATP is a universal energy storing molecule used by virtually all living organisms. In addition to its metabolic function, intracellularly, growing evidence suggests that release ATP into the extracellular space can serve as a signaling molecule, and so they explored this ATP sensors is with they insert the CBGFP with the TM5 and TM6 with the GPCR, and they optimized the linker, and also when they do this is with the hex cell, and then with the basal, and when add the ATP, and so the sensor first expression on the hex, and then add the ATP, and when the ATP is showed very good signals, when can see the cells working very good when on the cell, and then it also did with they also expressed this sensor in the different neural cells like astrocyte and neuron, and found that they when especially expressed on the astrocyte for this sensor, and it can detect the ATP have with a higher affinity. And with the neuron, it also showed good results, but it's lower, and is like so. We used this sensor that is specifically expressed on the astrocyte, and so for this, and then I want to introduce a part of our work in now, and then we found that the actual cellular. Oh, okay. The actual cellular purine signaling is crucial for microglia migration, and we know fighting for accident and is a traumatic brain injury. And so that is Dr. Gan's group found that microglia will be moved to the injury, and then used this the questions involved in how is ATP released, and then how is ATP censored by microglia So that is when we injected to the mice, and then we found that this is a different when pre-FLA and post-FLA is stimulation by the 2-photon imaging, and so then we will say that we will also do the connectization with the GFAP. GFAP with the sensor that is over the 90%, and also that is our data to show the microglia's migration, and that's activated the microglia. So summary, that is graph sensor are genetically encoded GPCR activation based different neurotransmitter sensor, and they would help a lot in brain research. And the second, FLA injury induced ATP releasing effective microglia's migration and their morphology change, and inflammatory cytokine release, and blocking inflammation show the protective effects by reducing microglia. And thanks our lab, and especially, and I want to thank Professor Juan Liu, and because it's to give and thanks my colleagues and Dr. Xu, Dr. Xiong, and it's very happy to with you. Thank you for your attention. Question.
Thanks a lot for Dr. Liu's excellent presentation. We have time for one quick question.
Yeah.
That's great. I got one question is like you did really impressive work on the a various of technologies. I just wonder do you have any plan to apply one of them or some of this kind of technology into clinical applications, either in short term or in long term?
Yeah, it's actually is for clinical. This actually is only for research to help us to make clear with the mechanism. And if actually we did this this sensor, it can work for a longer time. For a longer time, is more than 20 hours or 24 hours. And am I asking your question?
Yeah, great. Thanks a lot. Let's thank Dr. Liu again.